All right. Morning, everyone. So, you kind of see me over here, a little backlit. So it's my pleasure today to introduce Dr. Rashad Manangar. He is our extension agent here in Hawaii for invasive species. Rashad did his PhD at UH Manoa. He is an incredible expert on insects. He's doing work with the Rapidovia Death Project, Coffee Berry Borer. He's the go-to guy that whenever we have a question, he asks. And he's also friendly and nice, which is a great trait to have in a scientist and an extension agent. So he'll be giving a talk today on entomology, study of insects, and then also IPM, which is a term you'll hear a lot, integrated pest management. I think he's split into about 3 presentations, so we'll take a little break between them. Yeah. And again, for folks attending via Zoom, let us know if you have a question. For folks here, got to pass it out. If you have questions, write them down in the cards, and we can pick them up during break time and go over some of them.
Yes, there is an antibiotic.
Yeah.
Okay, anyway. So, thank you, James, for a nice introduction. Again, I'm an entomologist by training, but I do some, so I'm an invasive species extension agent, yeah? So invasive species have lots of pest, insect, and pest diseases, and pest weeds all are invasive. All invasive species,
Hey, James and Roshan, can I stop you for a second?
What's up?
We're having a little bit of problem with hearing you, and it's not just our end. We've got some chat that says it's not clear and a little bit muffled and echoed. So possibly closer to. If possible.
Yeah, Roshan, try saying something now.
Hey, Russell, can you hear me?
I can hear you. So how do you guys feel about that? Yeah, we're okay on our end. How about in the chat realm?
So, okay. Again, go ahead. All the pest species here in Hawaii are invasive species. So, by saying that, I will start my presentation. So I will be talking about general entomology. It's a kind of a 1 semester long course when I was a graduate student, but I will try to finish it in half an hour. So entomology is a vast topic.
Yeah,
We're all doing okay, I'll just take it out. Yeah. Okay. So entomology is a vast topic. It has lots of disciplines like insect ecology, insect morphology, insect taxonomy, physiology. And among them, IPM is a small portion of principles of pest control. So in these 3 hours or whatever hours, I will be talking about general entomology and one small portion of principle of pest control, which is IPM. It's called integrated pest management.
Hold on 1 sec, Roshan. There's still problems here. Try saying something again.
Can you hear me?
Oh, say a little more.
Hey, James, how are you doing?
Can everyone hear it?
Yeah, we're getting better. And I think maybe we just keep, we got some people saying half and half.
So maybe just put it here.
Okay. Yeah.
Now, hello, everyone. How is it now?
That's better. That's better. Yeah. We're getting lots of betters.
Okay. Awesome.
All right. Let's see if it works now.
So world of insects. So entomology is study about the insects, yeah. So worldwide, it is estimated 30 million of different insect species, and it is almost 90% of all living things in the planet. And among the insect, beetles account 1/4 of the whole insect world, different kinds of insect. And also 10% of animal biomass is of the world of ants. And 10% of animal biomass is from termite. And for every human being, there is 200 millions of insects in this world. So the world of insect is very vast. And this is the beetle, which occupies 1/4 of the whole insect world. So this is a very integral part of our organisms in the world, insects. So role of insects in the garden, some of them are pests, some are natural enemies, and some are only there just to sit around. So what are the pests? What this pest cause? So some pests cause direct damage by feeding, and some cause indirect feeding damage, and some are the vectors of plant pathogens. Some does only cosmetic damage in the fruits. They just make it, some fruits are edibles, marketable, but they cause some cosmetic damage. Some are the contaminants, they poop in the fruit or flower or in the leaves, so they contaminate the fruit. And some are the quarantine pests. Some are here, not in the Oahu, so they are quarantine pests. For just like now, coffee rust might be a quarantine pest for here from Big Island. And most of them are beneficial natural enemies. Also, most of the soil pests are soil conditioner. There are a lot of them, just like some of the mites. Mites are not insects, but mites, earthworm, rabbits are insects. Some of them are pollinators. Some recycle nutrient in the soil, and some just they just stand in the ecosystem and look around. So less than 1/10 of that is 1% of the insect species are serious pests in the agro-ecosystem or in the natural system as a whole in this world. Only 1% of whatever we have, 330 million different insect species, only 1% of them are serious pests. So, as I said, there's lots of insects, yeah? It's very difficult to do ID, but identification of insect species is very important to go forward in our business. In agriculture, we are mostly in agriculture or in natural resource management. The first thing is when we get some kind of problem with the insects or the disease, the first thing is to do the identification. Once we do the identification, then the door will open. The identification is the key. So once we do ID, suppose we get the insect, we don't know what it does, we go around, ask people, then we know like this is a mayflower and this is a June beetle. Then we have something to search about, yeah? We can find the scientific name and we can just Google out and there will be lots of information on one specific insect. So the taxonomy is the study of description of the insect, identification, nomenclature. We will go a little bit after that. So it unlocks the curiosity in us, and then we can go forward and find the information, and if we need to manage or whatever we need to do, we can go forward. So identification is the key. So as I say, taxonomy is the study, we do classification in study. So I compare classification of two organisms. One is us, human, the another is bee, yeah? So the classification has different labels, like kingdom, and both are animals, and phylum, as you see, arthropoda, they have a segmented legs in the insect, and we are chordate, that we have a vertebrate, yeah, backbone, spinal cord, and class, it's insecta, us, mammalia, we take care of our babies, yeah, feed, make them bigger and compete in this world. And in insect, order is the important. We study different insect with respect to order. So as if we work in the insect area, we need to know the insect order at least, then family. So if you are a graduate student of the entomology, the order, very good handle in order and family is needed. Then genus species will be mostly for the taxonomists. They will try to find out genus and species. For me also, if you ask, if you bring an insect and ask what is this, it would be very difficult to answer right away. So there are like 20 million insect species, yeah. So I'm only familiar with only a few of them, like maybe 100 of them, which I'm familiar with, which I have studied, or which I did up until now. I did some work on those insects. So there is an insect taxonomy that we can consult with, or there are some of the scientists who are expertise in some family of insects. So there is a genus and species. Us is Homo sapien, and this honeybee is Apis mellifera. So if we get to genus and species, so when we punch in the Apis mellifera in the Google, there is lots of information. So we can do lots of things if we need to study these insects. So this is also a nomenclature. We need scientific name, because I'm from a different country. I call this insect I know this insect with some other name. This is Rhinoceros Beetle, maybe in India. Somebody from India. It looks like India. Yeah, this is very common insect in India. Yeah, Rhinoceros Beetle. It's known by some different name here, Rhinoceros Beetle. So we need a scientific name. Like this is a Kamehameha butterfly. Yeah, native butterfly of Hawaii. Its name is a little bit difficult to read. Vanessa Tamiamia. So then the last one is the name of the author who described this species. So there is no bracket, yeah, no, it's not in parentheses. So that this species is described by this author, and it's stayed until now. For Rhinoceros Beetle, Oryctes rhinoceros, the L dot is the lineage, the name of the author, it's in bracket. So this species is first described by the lineage, and it is redescribed or it's repositioned later by some other people. So that's how it means. If it is in a bracket, it's real, the lineage described first in some other way, maybe genus same, species might be different. So then it's again redescribed by some other people, but lineage is still there. And this is a tobacco hornworm, Maduka maculata. Sorry, it's difficult to read these scientific names for me. Yeah, so Howard is the author who described this species. And again, in parentheses, somebody else has again redescribed or repositioned these insect in a different genus or species. Again, I was talking about the why we need scientific name. Yeah, so this is some kind of a water bug, giant water bug. I would say this is a giant water bug. So different people in different geographic locations say it or known by a different name. The water strider, water skaters, water schemers. That's why we need to know the scientific name of the insect. Yeah, and also. The bottom one, one is vinegar fly, the other is fruit fly. So normally what we call is it's a fly, yeah. So if we can reach up to, so fruit flies are all Diptera by order, and if we can go up to family, so Drosophilidae is the family, and Tephitidae is the family. If we can go up to family, it's very good information. So genus and species is difficult. So that's a nomenclature, how we name insect and how we know the insects by some common name or some scientific name and go forward. So this is about insect morphology, parts and pieces of insects. So as I said, insects are arthropods. Yeah, they have a jointed legs. That's why they are called arthropods. And its jointed legs are called appendages. They join with each other and it's bendable. And the body parts are segmented, yeah. And the body covering, the outer cover is called exoskeleton. It's a chitinous substance, And in insect, they molt in different stages. So they molt to grow. And these insects are related to spiders, ticks, scorpions, millipedes, and other crustaceans. As I said, entomology is very vast. So this is related to more insect evolution. They are evolved from other species like spiders, ticks, and scorpions. Again, their body parts can be divided into 3 regions: head, thorax, and abdomen, as you can see in the picture. So to be an insect, there needs to be 1 pair of antenna, 2 antenna, which arising from the head, 3 pairs of legs, 6 legs from prothorax, thoracic region. All the legs are from prothorax, and it should be 2 pairs of wing to be an insect. So 1 pair of antenna, 3 pairs of leg, and 2 pairs of wing for an organism to be an insect. So same thing, as you can see, there is a head, thorax, and the abdomen. It's for honeybee. And also this is a Staphylidae. I don't know the name of the insect, but family is Staphylidae. Same thing. You see a little bit different structure, but there is a head, thorax, and the abdomen. And elytra is the portion. There is no wing, yeah. So elytra is the modified wing. And in the larvae of the tobacco hornworm, the larvae can be also, so those portions will form as a head, abdomen, and thorax. So see the top one here. It is head, thorax, and abdomen. Again, so counts the leg. There is 6 legs, 3 pairs of leg attached in the thorax. So the different pieces and parts, appendages of legs, different names. Coxa, femur. This is just like our hand, yeah. Coxa, femur, tibia, something like a. And so some insects are wingless, like a silverfish, fleas, and some termites and the ants. The termites and ants will produce wings when they need to mate. And in Diptera order, there is only 2 pairs of wings. In like flies, this is a hoverfly. There is only 2 pairs of wings, and another second pair is modified. You can see this small structure, yeah, modified to become halters. So the main function of the halter is to balance their flight while they are flying. The 4 wings will help them to fly, and this halter, the reduced wing in the form of halters, will make them balance when they are flying. So in beetles, the 4 wing is hard. So we just saw the elytra on the Staphylidae, yeah? Hard elytra. And the hind wings, the second pair of wings, are membranous. So the hind wing will help them in flying, and this one is more of like a for the protection only. So that's the reason why the beetles cannot fly for a long distance. So different kinds of antenna. I just put only 3 to 4 4 different kind. Also, I think there is 12 different kinds of antenna. So filiform, which is just like in cockroach. So this is a pectinate. It's very nice antenna. See, you can see, yeah. In the click beetles, plumose. If you can see the mosquito in the microscope, you will see this kind of antenna. And in most of the flies, you have a very short antenna. It's called erictate. And mouth parts, mostly 3 different mouth parts. Chewing mouth parts, just like in beetles. Their mouth part is just like us, the upper lip, lower lip, mandibula, something. So labrum is the lips in us, and mandible is these pieces coming out, yeah, is the teeth. So they broke the pieces, the big pieces into small pieces, and they intake and chew, grind, yeah, then eat. And sucking mouth part, this is mostly in the moths and butterfly. They have a proboscis. Actually, this proboscis is the long tube. It's when they are not feeding, it's coiled and placed underneath the mouth. So when they need to feed, they uncoil and extend and suck. So, mostly they suck the nectars and sometimes pollens. So it's mostly in the butterfly and moths. And another is in a piercing and sucking. It's called stylet. Stylet is also it's mostly when they are not feeding. It's like ventrally situated under the body, and when they need to feed, they extend the stylet and pierce in the plant tissue and suck the sap. It's mostly in the aphids. We'll see now. So this is how the insect develop from the egg to adult. There's two different kinds of development process. It's called metamorphosis, and it's two kinds. Yeah. Simple metamorphosis and the complete. In the simple metamorphosis, the stage of insect doesn't change. Just like an egg hatch into babies, and the babies grows in size and become an adult. In complete metamorphosis, the stage or the forms of stages will change drastically. Egg will hatch into larva, then larva will develop, grow in size, they complete different instars, and pupate for some period of time. They can pupate for as long as overwintering or 6 months, and then become adults. So simple is they just hatch into babies and grow in size, become adult, and incomplete, they change their forms. Eggs into larva, larva into pupae, and pupae into adults. This is just an example of what kind of metamorphosis? Simple. So they hatch into babies and grow in size. This is a leaf-footed bug. We don't have this bug in our island, maybe not in Hawaii. See, you can see this foot. It just looks like a leaf. So that's why it's called leaf-footed bug. So eggs will look like a barrel shape. It's just laid in the line. They hatch into babies. Babies grows inside. Each time they molt and grow in size. So maybe they molt 3 times or 5 times. I don't know how many times there is. So they molt and grow inside and become an adult. So this is simple metamorphosis. Nothing happened. Yeah. So these are some of the orders with the incomplete metamorphosis. Incomplete metamorphosis is the simple metamorphosis. They do not change. Yeah, fleas, termites, grasshoppers, true bugs, aphids, and the hoppers. So we always call bug. Yeah, what kind of bug is this? Somebody is bugging me. Bug is, so what is bug? Yeah. So normally the what do you call it? Pentatomidi, like this kind of, we call this is the bug. Green stink bug, whatever other bugs. What kind of bug we have? Bug. So this is a bug. In entomology, this is the bugs. So hemipterans are bugs. So what is really, every bug is an insect. Not all insects are bugs. Only true bugs are bugs. So this is the true bugs in the order of hemiptera. So to be we call stink bug is the bug. Yeah, it's a true bug in the order. To be a bug, we need this prothorax, the triangle-shaped structure in a prothorax, which is called scutellum. And as I see, so these third of the wing needs to be clear. You see this one, yeah? Third of the wing needs to be clear. Then it is bug. All insects are not bug. Only this is bug. It's in the Pentatomidae family in Hemiptera order. So these are the bugs. See? in a plant bugs. It's in the Myriad family. Seed bugs in the Lygidae family, and this is the assassin bug. This is also a bug, true bug, not general bug, true bug in these families. Pentatomidae is the one which I show you, this one, and these are the other families. So these are homopterans. Homopterans are, homo means like a kind of, I don't know, it has a name, but it's related with the roof-like held over. Their wings are roof-like and held over the body. As you can see, these wings needs to be like a roof. This is one of the feature of homopteran, but not this one. So they are a sucking insect. They suck the sac. And another thing is their wings needs to be membranous. So different kinds of hopper, leaf hopper, plant hopper, tree hopper, cicads, aphids. Aphids are also homoptera, xylids, and whiteflies and scaled insect. Do you know what is this? This one? This insect. Even I didn't know this when I saw it first time. So that's why I'm asking. This one, this is a bug. This is a regular bug, not true bug. So when I started this job, I got this sample, came in the extension office, and I don't know what is this. So this is a scale insect. It's called barnacle scale. So if you see, this is the pest of mostly shrubs, ornamental plants. So if you see this, this is also a bug, regular bug. It's a scale insect. What I see, Orthoptera. Orthoptera means again the Orthoptera in the Orthoptera hind limbs are longer than the four legs, 2 forelegs. So that they can jump. Yeah, these can jump. They don't walk. Grasshoppers, crickets, and the katydids, they don't jump. So they can jump. That's the main their feature. If you see jumping insects, they are from Orthoptera orders. And another thing they do is they lay eggs in the tube. So if you have a leaf and they lay egg in between the lamina, 2 lamina of leg, they insert eggs in the lamina. If you see, that's also very fascinating. If you see in the field, the eggs will be in the leaf lamina, in the margin. So that's the grasshopper egg. And they communicate with making chirping sound. So we talked about incomplete or simple metamorphosis. So this is a incomplete, no, what I said before. Simple. So this is complete metamorphosis. They change their forms. Egg hatch into larvae pupae, and then into adult. Yeah, this is the ladybug eggs. The ladybug larvae looks like this. These are mostly predators. The larvae is the predator. We know ladybug is the beneficial insect, yeah, but larvae is the predator. They feed on other insect egg, and the other soft-bodied insect they can attack. They pupate like this, and the adults. So these are the few orders of insect that undergoes complete metamorphosis, like Coleoptera, which is beetle family, one of the largest family in the insect world, Diptera, Lepidoptera, and Hemiptera. I think it's Coleoptera at the top.
Yeah,
So, huh?
Yeah, we press the Alt key.
Huh? Alt? Okay. No. So for this, removing this. Okay, good. Thank you. Just a general description of, so we were at the Coleoptera, yeah? Coleoptera, different families of Coleoptera. I talked about Staphylinidae before, Curculinidae. They have a, the mouth part is, I don't know what do you call this? Like an elephant something, yeah? So different families, Staphylidae, Curculionidae, Carabidae. The Carabidae is another beneficial insect. They live in the soil. Chrysomelidae, Coccinellidae, and the Scarabidae, different orders. Just to Diptera, again, flies. We talked a little bit about flies also. Yeah, one pair of two, it needs to be two pairs of wing, but we will see only one pair. Another pair is reduced to form a halter. In mosquito also we see only one pair of wing. So Lepidoptera, we talked about siphoning mouth part, yeah, which stay coiled, and whenever they need to feed, it will uncoil and feed in the and feed in the whatever. Some examples of some families of Lepidoptera: Saturnidae, spinged moth. This is a Puridae, cabbage butterfly. You will see in the cabbage field in the daytime. Monarch butterfly, and I think this is a silk moth adult. Beautiful, yeah. You can see some samples in the insect box also. Neuroptera, this is we normally call lace wings. The most distinguishing character is the what do you call it? Net vein wings. As you can see, the wings are transparent, and you see the networking wings in this family. These are also predator. Larvae is the predator. Thysopneptera trips. So trips, one trip is also trips. Trip is also trips. Trips is so in trips also different families of Trippidae. I don't know how to say. So these other kinds are also predatory in nature. Most of the trips are pests. Some transmit diseases. Some are the vectors of plant disease also. And these two are predators. So the most unique thing in the trips is their wings are fringed. There is a one main, what do you call it, rib, and so the wings are fringed. If you, I don't know how to describe. Anyway, okay. Hymenoptera, another important order in the insect world, which has these main families. Formicidae, most of the ants. Vespidae is the yellow jacket. Apidae, all the honeybees, bees and bees. Ichneumonidae is a parasitoid wasp. Beneficial insects. The one distinguished feature in this order is they have a narrow waist. In all cases, they have a narrow waist. So this is the general entomology. A little bit morphology, a little bit taxonomy. We try to dig up until to the family level. And yeah, as I said, this is a 6-month long course, and we finished in how many minutes? Anyway, so these are also pests, but not insect. For spiders, for organism needs to be insect, they need 3 pairs of leg, yeah? So the spiders had 4 pairs of leg. So they are not insect. Similarly, mites, we have a mite pest. Mites has also 4 pairs of leg. Centipedes has lots of legs. So these are also pests, but not insect. Nematodes, again, millipedes. These are the, I think, symphyllins. Roly-poly, these are called, I think, roly-poly. Yeah. So these are not insect pests, but they are pests. So as I said, the key to entomology is the identification. So we get to see lots of insect in the field. Maybe this is insect, only, as I said, only 1% of them are insect pests. So the main thing is ID. So for the identification is but difficult. As I say, there is 20 million insects, and getting known to around 20 or 30 is also challenging. Yeah. So we have resources. CTAR has a resources. It's called, if you go online, extension into something. So you might need to explore a little bit, but it is called Knowledge Master. So you go to crop Knowledge Master. You can see by crop or by insect. There is information. It's old but still useful. I sometimes use it. It says what is IPM, IPM program. So if you explore, there's lots of information. Maybe it's old, but it's useful. The another thing I use is IPM images. You can register in this website, and there will be it's a photo identification. You can get some kind of information with the photo, and you can download the photo. In most of my presentation, if you see this number, UGA, that's downloaded from IPM images, UGA. So this is also some kind of resources for insect hiding. And another is a CTAR publication, extension publication. Oh, sorry. CTAR extension publication. There's lots of publication on insect diseases and all other kinds. So these are the infections you might need to explore if you. So another thing I use is Scott Nelson is our retired faculty. He's a plant pathologist. He has a Flickr account, which is still. So there is lots of information in this Flickr account with the Scott Nelson. So what you do is you go to this search button, yeah, here, this search button, and you enter whatever you like. Papaya disease. So there will be like 500 of papaya disease photos. If you punch in Carlo diseases, there should be some kind of 20 or 25 photos. So lots of information in this website too. And I use this one too, Bug Guide. So other than that, so if you have some questions on insect ID, you can contact me. So I might not answer, but I will try to find the answers from some other folks, some other resource person. The person which I normally contact is Dick Suda. He's an agricultural diagnostics ADSC. He's an insect guy in the ADSC at the US Manawa. Another is Janice Masnaga. He's a taxonomist, insect taxonomist at SGOA based on Oahu. Maybe you can also reach directly to them, or you can contact me if I can. If not, I'll find the ways to ID the insect. With that, we finished our first presentation. Thank you. Thank you for listening. Any questions?
All right, we've got one question here. Someone says, I often see millipedes and symphylins at our planting site. What types of damage do these cause? And symphylins are the roly-poly.
Symphalids are roly-poly. Symphalids are, I think I would say, symphalids are beneficial insects. They help in nutrient recycling. If there is a symphalids, roly-poly on your soil, the soil stinks, yeah? It stinks very bad. That means there is some kind of nutrient recycling. And millipede, I don't know. They might not be pests.
They're pests to us.
Pests to us. Millipedes, centipedes are pests to us. Maybe they are also related with the soil organic matter. If there is a high organic matter, you will see these kinds of insects, roly-poly, centipedes, millipedes. I think, yeah, maybe I showed you as a pest, yeah, maybe not pest. Just a stands still, yeah. Thank you.
Other questions here? Questions from online folks? I see a question about the bug guide site. I'll put that in the chat during the break.
Oh, lots of questions in the chat. What was the bug guide site? So how do I? Oh, can you please the curtains? Okay, we'll take a break and do that stuff. So bug guide, you can Just punch in the Google, it will come up, I guess. Good morning, a scale. Can you please close the curtains? Yes, we will. Let's probably join.
I think that was all the questions, Rashan.
Okay, thank you.
Thank you.
Yeah,
That's really good.
yeah, you're welcome.
Take a 10-minute break. I
Okay,
think we're doing okay.
yeah.
So it's around 9:40, so around 9:50.
So we will come back at 9:50. Okay.
Okay, sounds good.
Maybe you and I will switch places and then the light. Yes.
Probably turn on the overhead light. It might eliminate it a little bit.
Still. Well, it's with words.
All right, I think we're coming back. So we'll start out with Rashawn's second talk.
Okay. Welcome back again. So, again, small portion of principles of pest control, IPM, integrated pest management. So, when I was a student, we need to remember definition of IPM, yeah, to write in the test, or they will ask, define IPM. So, in a graduate school, when I explored a little bit on IPM, I found 68 different definitions of IPM, many definitions of IPM. So I studied a little bit, only a few definitions, and tried to summarize definition of IPM and put it in here, this slide. It is an ecosystem-based, needs to be sustainable. It needs to give a long-term prevention, that it's effective for first 2 years, just like a resistant variety. We develop resistant variety, it's effective for 3 years, then it loses resistance. It's not sustainable. And we normally use combination of techniques. If we can, we don't need to use all the techniques like 2 to 3 combined together. If we can, if it's available, we need to combine one or more techniques to control insect, and it should be economic and socially accepted. It's not like we don't grow GMO papaya here, yeah? It's not socially accepted, maybe. So it's not IPM. So all these 4 criteria needs to be there to be integrated pest management. So the most short and sweet definition of IPM, which I like to tell people, is a decision support system to keep pest population below economic injury rate. It's a little bit technical, but I like this definition because we decide when to do what. I don't tell any people to do this, because I do tell them, maybe they cannot do it. A grower or a person who is managing his crop, he will assess his problem, he or she will assess his problem and decide what to do and what's available and what he can pick to control that insect, to put the insect population below some limit and harvest economically. He needs some product. Yeah? So it's a decision support system to keep pest population below economic injury level. We will talk a little bit about these technical terms, economic injury level, economic threshold level, also later in the slides. So a little bit about history of pest management. Traditionally, before 1938, it's a kind of traditional approach. The Chinese people, starts with the Chinese peoples using most of the oils and the botanicals. Then starts 1939 to 1962. It is the era of pesticide when DDT was discovered. It's organochlorine. Then organophosphates, lots of insecticides were used in the wars and in the agriculture sector also. Then it came to some point around early 1960 when they found some residues of DDT in the milk. So that's the time when this Rachel Carson published this book called Silent Spring. From then onwards, the people became aware of the hazardous effect on the environment and the human beings of indiscriminate use of pesticide. Then after some period of time, there is a professor called Robert Van Den Bosch. He published another book called Pesticide Conspiracy. So this is just like something which we don't want, but the pesticide company want. They like to produce pesticide, one after another. Yeah. So he is a proponent of integrated pest management. He put the concept of concept of integrated pest management around that time, and it was from there onwards it was implemented and popularized. For now, it's a little bit kind of era of GMO, and maybe it will be in the future also. So just a little bit on some of the terminologies. What is pesticide treadmill? Aligning with this pesticide treadmill, there is another 3 terms, which is pesticide resistance. So what we do is we spray pesticide for some insect problem. Suppose we have 100 insects. Yeah, we spray some specific pesticide. We kill maybe 97 of them, and the 3 will escape from the pesticide. And that 3 insect will multiply, and in next generation, there is some population of pests. And we again spray same insecticide. So maybe only 50% of them die. So the 50% left will develop resistance in the 3rd generation. Around 3rd generation, the pest becomes, when we call, in the 3rd generation, we will kill only 10 of them. Out of 100, we will kill 10 of them. So then we say the pest has developed resistance. So by spraying same class of insecticide over and over, what we do is the insect will develop resistance, and we cannot kill the pest with using same insecticide. The pest rebound means normally the insect pests and their natural enemies are in equilibrium in our ecosystem. So the pest population doesn't drastically go up unless we do something, we spray pesticide. What we do is while we use effective pesticide, we kill pests as well as natural enemies. So then after some time, as I say, some population becomes left out or become resistant. Yeah? The pest population increase, but the natural enemies population cannot rebound in the same scale. So that there will be even more insect pests than before, because we removed natural enemies, yeah, and they could not establish, they could not rebound as up to intensity to the pests. So it's also called pest resurgence. There is more problem with pests than before. So another thing is called secondary pest outbreak. So what normally there is lots of insect pests in a crop. One primary pest, lots of secondary pests. Secondary pests are not problem. We have only, we wanted to kill only primary problem, primary insect, primary pest. Secondary pests, they stay there, they do a little bit of damage, but we don't care. When we spray, what we do is we might kill the natural enemies of secondary pests. That's what they are, secondary. We kill the secondary pest, we kill the primary pest, we kill the natural enemies of secondary pests. Then in next generation, primary pests are controlled. We eliminated natural enemies of secondary pests. Then the secondary pest becomes a primary pest. So there are 300 most damaging insects here in the United States, which are secondary pests. There are secondary pests before, and they become primary pests later on. So I can give some examples later on, but these are the scenario of using a pesticide in an indiscriminate manner. So we call it pesticide treadmill, that there will be one kind of pesticide we use, develop resistance, then another new pesticide will come up, use, again resistance. So the growers will totally depend upon the new pesticide all the time. So like a pesticide will control grower. Just you change pesticide all the time. So these are the few steps. So that's why we need IPM. Yeah, it's a decision support system. We need to decide. We need to be a little bit credible, not depend upon pesticide. So these are some steps in the IPM, the identification. So we talked about identification is key. And we also need to know about the damage symptoms, how the pest damage, they choose the damage symptoms of pest. And we need to know the pest life cycle, like we talk about, a little bit talk about the metamorphosis life cycle and the biology, and action threshold. Again, I will go to another slide to describe action threshold. Then, so we know pests, their damage symptoms. Then if this pest is a problem, we need to monitor their population. We need to know population dynamics of the pest to set the action threshold. And then after that, we need to choose and apply combination of management tactics. We need to choose whatever we can do, yeah? Whatever is available. Then after doing that, we need to evaluate the result. That's the steps of IPM. So you can see this graph. In the x-axis, it's the time, and in y-axis, it's the number of pests in a given environment. And these are the pest population. When we grow crop, the number of pest population gradually increase. If we don't do anything, it will keep on increase. So in a dotted line, and we have a economic threshold, ET. So this is the ET for some specific insect. It's a population density of a pest at which we should take a management action, ET. So at this point of time, we should do something to tame down the population. Otherwise, it will keep on increasing. Yeah? And EIL is economic injury level. So it is the pest density after which we will get the economic loss of our crop. Pest density that will cause yield losses or damage equal to management cost. So at this point, the damage is equal to management cost. So normally what it's indicating is below ET, we don't need to do anything. And after ET, something needs to be done. And at EIL, damage is equal to management cost. So if we do some technique of insect control above this line, benefit will be more than the cost of doing management. And if we do something below the EIL, mostly we will lose the cost is greater than benefit. So determining ET is very crucial, but it's very difficult. So we talked about economic threshold, yeah? If somebody will ask me, what is the economic threshold of diamond back mark? No answer. So it needs lots of research. It depends upon the places to place crops. It depends upon the crops as well. So it's a very difficult question. So as a grower also, they need some kind of experience growing crop and studying a population of some particular insect in their own environment. So I just came up with this disease also because lots of people also ask me to tell about some kind of disease also because it's raining, yeah, there is no more insect outside, maybe lots of disease now. So it's a disease triangle. So if there is a susceptible host, we have a pathogen in this kind of weather. If there is a susceptible host, favorable environment, and the proven pathogen, so we will see disease. So how do we manage disease? If we break up one of these links, that's also IPM. If we were able to link, so we can make susceptible hosts into resistant. We can make a not favorable environment, just like so suppose rainy weather is favorable for disease incidence, yeah. So mostly in the rainy weather, soil is wet. In most of the cases, soil is not well drained. So if we plant the crops in a well drained soil, that will be deleting a link of an environment, favorable environment, so that the incidence of disease is less. Okay, so as I said, we have an insect problem and we need to do IPM. So first thing we need to do is a scouting of pests or scouting of something, damage symptoms or pest. So normally scouting is a systematic approach to assess crop health or pest outbreak without examining every plant. So we don't need to examine all the plants in the field. We just sample. So it relies on sampling a subset of plants in the entire field and the collected data periodically. So this is one of the steps of setting the economic threshold. So this is a photo from Texas Agri Life. That's how they scout their field. This is, what do they call this one? Drone. So they fly over the drone in the entire field and they look at the picture of their field. I think they have some kind of hue and chroma combination. So mostly yellow fields are nutrient deficient or the disease field, and green fields are good field, but undamaged, nice field. So they again go back depending upon the hue and chroma of the photo that's taken by the drone. They go back to that place and try to find out what is the main problem, either disease, fertilizer, or insect. That's applicable for a larger area of field. So that's one new technique of scouting, but we don't have to do it here. And the main objective of scout is to find out what's in the field, as I said. Either it's for us now, it's a population dynamics of the insect, or the severity of disease symptoms, or to find out how diseases are progressing. Like this week, some patches of disease are here. Next week, it growing in size, a larger patch. So, as I said, for scouting, we take sample. Yeah? So for insects are not uniformly distributed. We found insects often in patches, mostly in the edges. They start with the edges, then gradually go inside the field. So what we do is random sampling. We take sample randomly. So normally what we do is if it is a field is larger, we do a transect in a zigzag manner and a sample in some specific point. So we also take sometimes we also take a systematic sample, which is called purposefully taking damaged sample or unhealthy sample, because we need to find out what is the cause of those damage. Yeah. So sometimes we also take systematic sample. It's mostly in the scenario of taking sample for the disease plant. So what would be the sample size? So greater is better, but we need at least 10 to estimate some kind of density or some kind of disease evaluation. Again, sampling frequency. Frequently is better. If you can do 3 days interval is better, but 1 week or once in a 10 days is also okay. So the sampling methods for insect, we use sweep nets for some fast moving insects, and like a leafhoppers, grasshopper, and butterflies. And also beating cloth. What we do is we put the one cloth underneath the plants and shake the plants. These are all photos from Texas Agri Life. And also, if it is some kind of a shrub, what we can do is we put the beating cloth and we put the beating cloth underneath the plants and beat the plants, so that insect will fall down and we collect the insect. And another method is plant inspection. We inspect plant, some specific leaf, we can standardize depending upon our experience. If we think the insects are mostly in the newer leaf, we sample newer leaves. If it is in the old leaf, diseases are in the older leaf. Yeah, so we better sample older leaf. If we think new leaves are too new, we cannot take it out. We can sample 5th leaf, 10th leaf, or that's based on experience. So we sample, then we observe through eyeglass. Sometimes we bring it back to lab and observe through microscope to count the insect. So another sampling tools, this is specific for some insect species, lots of traps. This is called, the first one is called standard New Jersey light trap. This is for monitoring mosquito. Pitfall traps, monitoring ground beetles. Burlesy trap, this is for also soil insects. mostly mites, soils, and mesoarthropods. What we do is we sample soil and bring it back to lab and put it in the, this is the first, put it here. And what we have here is on the top is a light. So with the lights and some heat of light, the insect will crawl down in this container with the alcohol. Then we collect and observe to microscope. Again, different kinds of traps. The Japanese beetle trap, this is mostly used for Japanese beetle and other beetle. Inverted funnel trap, especially for moths. Multi-lure trap for fruit fly. And so this is, I think, one example of a test scouting in the Hooker pig field. As I said, it's a random sampling. Yeah, we do the transact in the zigzag manner, and we take the sample in these points. The W set is mostly convenient. So zigzag manner, we take the sample in these points. So in cucurbits, what we found, the most key insects are aphids, whitefly, thrips, fruit fly, and pickleworms. So, so if I need to take a sample of aphids or count the aphids, what I do is I go to this specific plant and select a leaf and turn the leaf and count the aphids and take the data. For whitefly, what I do is, this is an entomological method. For sampling for whitefly, what I do is I take the leaf ticks sample. There is a cardboard. I just take a leaf ticks from somewhere in the plant and bring it back to lab, and I count eggs and the meat for whitefly. And for the thrips, I take leaf sample and the flower sample and bring it back. For leaf sample, what I do is I will wash the leaf in the alcohol, 70% alcohol, filter to coffee filter, then count the thrips in the coffee filter. And for flower also, I bring the flower and dissect the flower and count the thrips. Fruit fly, trap catches with the lure. Pickleworm, again, flower sample. Sample flower at the flowering time. And bring back to lab and count the eggs of pickleworm underneath the microscope. Difficult, yeah. So this is a part of my study during my graduate study. So what I did is I counted thrips in the leaf, in the cucurbit crop. So x-axis is the days after planting, and y-axis is the number of leaves. Just ignore this blue line and the green line. And this is the population density of thrips in the corn monoculture, not corn monoculture, cucurbit monoculture without any intercrops. As you can see, as I show you the graph of that economic threshold, yeah, this just looks like the same. The population gradually increase, and which is big, and the crop health will go down, then the insect will go down. So my concern is, how do we estimate economic threshold here? So we need this kind of data to set up and to find out the economic threshold. This is the 1 year data. Maybe we might need 2 year data and try to find out the damage also at different level. Then only we can find out economic threshold for this thrips in the cucurbit crop for Oahu. This is done in Oahu, yeah. So for now, I would say this is gradually increasing, and this here at some point the increment is a little bit more higher, exponent, not exponent, the kind of exponent. So what I say, if there is a 25 around this area, 25 trees per leaf, it might be economic threshold, but we cannot conclude. We might need a 3 year data to tell this is the ETL and some kind of damage. Maybe at around 25 trips per leaf, there is a damage caused by trips. Just an example how, if we need to find out economic threshold, how we can do, but there is no answer. So how do we evaluate disease? So in my again, when I was a student, I did some research in a corn disease vectored by corn plant hopper. It's called Maize mosaic virus. These corn plant hopper cause hopperborne. Feeding damage is called hopperborne, and they also transmit Maize mosaic virus disease. So this is a hopperworm symptoms, only feeding damage. This is a maize mosaic virus. So they also cause virus disease. So to evaluate these, this is what I did is I have several rows of corn and I selected 4 rows. And in the 4 rows, I counted plants with the hopperworm disease. And this graph came up like this. Again, in the y-axis, percentage of hopperworm symptomatic plants like this. So which is higher at when the plants are small, higher, and it gradually went down. And so hopperworm symptoms went down and the virus symptoms increased. So there is a feeding damage. To transmit the virus, the plant hopper needs to feed and inoculate the virus, yeah? So there is a feeding damage at the beginning, and the feeding damage was more at the beginning. This went down gradually. Then after feeding, they inoculate the virus, then the symptomatic virus plants increased. At the end of the season, it's almost 100%. Like all the plants are symptomatic at the end. It's just to show how to evaluate disease. So again, we are around IPM, ET. So just to decide, we need to decide when to do what, yeah? So this is the sampling plan of banana aphid. Banana aphid transmits banana punchy top virus. So this was around the early 2000s. This banana punchy top virus was a hot cake at that time. So lots of research was focused on banana punchy top management. So around this time, so lots of researchers don't know when to do what. Yeah? So they're trying to find out economic threshold, or what time they need to do spray. So counting aphids in the banana plants is very difficult. It's just a tall tree. There will be lots of one banana and lots of small keikis. Yeah. So this is a research done by Robson. Her name is Jackie Robson. Yes. So Jackie Robson. So what he did is counting banana in a tall plant. The mother plant is very difficult. So he chose small plants, keikis, and he has a cigar leaf as one and other leaf, 2, 3, 4, 5, 6. So what she found is the most of the banana aphids are concentrated on the 6th leaf, as you can see in this graph, or 7th leaf. So what she suggested is if you need to sample for banana aphid, you just you select them, because tall plants are difficult to sample. You select keiki and you look at the 7th or 6th last, or one leaf before that leaf. So things are now easy. You don't need to count aphids in the tall plant, mother plant. You select keiki and you just look at the last or the one leaf before that one. And she developed binomial sequential sampling plan. Binomial is only yes or no. You don't need to count. So because if you see aphids here in the 7th leaf, there might be hundreds of aphids. And it doesn't make sense counting like a 500 aphids, 1, 2, 3, 4. People may confuse and you lost the count. So it's binomial sequential sampling plan. You just look, yes, you record yes, no. So it's sequence. And you keep on sampling. So how far you keep on sampling? So it will be described by this graph. A little bit difficult for me also to describe. So what she found is you need to sample at least 20 banana keikis. So 20 banana keikis. And if you don't find any aphids, you are done. Don't treat. You are in this region, yeah? So at least 20 plants. If you don't find any aphids in those keikis, don't treat. You are done. No need to sample. No need to treatment. If you sample maybe somewhere 25 plants, 25 plants here, yeah? And if you get 2 plants with the sample, somewhere here, my arrow, yeah? 2 plants with the aphids, then it says this model will say keep on sampling. So you need to keep on sampling. So you keep now 30, you sample 30 plants. If you are in this region, only one plant, now you already have 2 plants. If you sample 30 plants and you have 5 aphids, here 5 plants with aphids, so it suggests you need to treat the entire field. If you are in this region, it says don't treat, and you don't need to sample anymore. If you are in this region, so at least 20, start with 20. If this says keep on sampling, you need to keep on sampling. And when eventually, if you are in this region, you need to treat your sample. Yeah, treat your field, entire field. So that's how this is a decision management system. So a little bit scientific, but at that time it was useful. Maybe not now, because a little bit older, or people may have their own methods to decide when to treat the banana field to control BBTD. So normally it's a control of aphids with the imidacloprid, and imidacloprid is recommended to kill the aphids and to control banana punchy top crimes. So just to give an example how the economic thresholds and the decision management system is developed, it's not easy. So again, sampling plan for coffee berry borer. We had coffee berry borer around like 6 months ago in September. It was already in most of the island has coffee berry borer. So I also bring the sampling plan of coffee berry borer as a component of IPM. So this one, most of the coffee growers in the Big Island do. We don't have that kind of problem until now. So you will see the holes in the blossom end of coffee cherry. If there is a CDP damage, you can see 1 hole and also 2 holes. So sometimes you can see the coffee berry borer entering in the coffee cherries. And these are the coffee cherries with the fungus. So, okay, let's go to another slide. So for the sampling plan of coffee berry borer, we need to maybe this EMG talk will have a Andrea Kawabata sometime later. Okay, okay. Now, otherwise I can talk a little bit. So these are the coffee stages in terms of coffee berry borer infestation. It's called A, B, C, D. So A stage is the green, mostly it's a green cherry. So coffee berry borer just started to enter in the blossom end of the coffee. And it's almost in, half of the body is in. It's a B stage. And C stage, the coffee berry borer adult is inside. And D stage, they already made a gallery. So normally it's called AB live. These 2 stages are AB live, where we can do something to control coffee berry borer. Once it will be CD, it doesn't call live now. Control measures are not effective. Any kind of spray is not effective. So for coffee berry borer IPM, they form a 33 sampling. So if you have a 22.5 acres of coffee area, you need to sample 30 trees. So what you do, so if you have a larger acre of area, you need to subdivide your plot into 2.5 acres. And you take 33 as a sample in each 2.5 acres. So in each 2.5 acre, you do a transact in a zigzag manner to sample 30 trees. So you go to first tree, what you do is you sample a lateral tree branches with around 50 green berries, and you count all the green berries in that branch, and also infested berries with the AB infested berries. You sample lateral branch with around 40-50 green berries. You try to find out percentage infestation. For that, you need to count infested berries and the total number of berries. Then you find the percentage infestation. And out of those branches, you collect the infested berries only, not all the berries. From that sample, first tree, you collect infested berries only, and you go to 30 trees. You repeat this in all 30 trees. And at the end, what you have is the coffee cherries or berries with the infestation only, with the AB line. No, with the infested coffee berries. At the end, you have an infested coffee berries from 30 trees. And you found the percentage of AB line. Out of all infested berries, how many percent of they are AB line, A and B? So we have two parameters. One is percentage infestation and another is percentage AB. So they come up with this table. So here's in the top, percentage AB, in the side, percentage infestation, and they have lots of different colors. This is some kind of economic analysis done by anyway, our CTAR faculty. So if you are in a white region, it says the values are 0 to 0.99. So if you are in this region, spraying is not recommended. You can read later on. So blue, there is a recommendation also. Another blue, 2 to 4.99. So at this point, it says, especially early in season, this is critical level to start spraying to avoid economic loss. See, so this area, when the values are from 2 to 4.99, this might be ETL for the coffee bean. At this point, spraying is recommended. And after this point, like in orange, it says you are losing money due to CVB damage, but you may still want to spray. At red, what it says is processor may reject your harvest. The value of harvest may not cover your picking costs, so consider focusing on next crop. It says you just wipe out your coffee at that time. Just harvest and throw away somewhere. Strip pick and stump prune. See, strip pick and stump prune. So that's how the principles of IPM will work. So by implementing IPM, we understand population dynamics of pests. We can determine ET in our own condition. We don't need to follow those things. We don't have to develop, but we can develop our own also. Then what we can do is we can also predict the population in next season also. At some point, I said we need to learn the damage symptoms as well. Yeah. So if the plants, if there is a chewing of leaves, stem, and fruit, if there is no insect and we see the only damaged plant, we can also come to know that if there is chewing of stem, it might be grasshopper, beetles, caterpillars, or slugs. So these insects will suck the sap. You see, leaf miners, weevil, twig borers, root borers, caterpillars will bore the leaf tissue, or bore the leaf tissue. Yeah, leaf miners will bore in between the leaf lamina. Yeah. So these are the symptoms, like damage symptoms with respect to these insects. If we see these kinds of damage symptoms, we can predict the insect might be these. So I will have a few examples of insect with their damage. These are flea beetles. We talked about this family also, Rhinoceros. These are the small insect. This is in the eggplant. You see, if you touch this insect, it will jump. That's why it was flea beetle. So the damage symptoms you see lots of small holes. So if you see lots of small holes in the eggplant, beans, and lots of other crop. I can remember only eggplants and bean only. So it's a flea beetle damage. So spider mite. You will see the spider mites are mostly underneath the leaves, and what the damage symptoms is, it's called speckles. These small dots are also called speckles. It's a bronzing spot. It's a brown, small spots. So if you see this kind of a damage symptom is mostly Spiderman damage. So so the thrips has a scraping mouth part. So what they do is they scrape the leaf tissue. So mostly you will see similar kind of damage in a mite as well as in thrips, but this you will see a spot, small bronze spot, but these are in the patches. What they do is they scrape the chlorophyll. Yeah, so that's how it looks like. You see. So another thing, idiophyte mite is very common here on Kauai. Lots of samples come into our extension office as well. I learned here after coming to Kauai, this is a idiophyte mite. Lots of galls. If you break the galls, you see under the microscope, you only see the mites, otherwise nothing inside. No inside, I don't know, something like this. So break the gall and look through the microscope and you see the idiophyte mite. So beetle damage, mostly they mix a small hole. This is a oriental sour beetle. So we have another beetle, which is most complained. Japanese beetle? No, huh? Chinese rose beetle, which comes after the ducks, and they come in a group and they defoliate everything in a night. And next day you go see like these symptoms. This is another beetle. They bore into wood and make a tunnel like this. Mostly ambrosia beetle does that. Do I have a movie here? Looks like
Try clicking on it.
no. I see.
Try right click.
So in this movie, I was showing how beetles will defoliate in a short period of time. But
We'll put the videos up on YouTube since we're having some technical difficulties.
there's some beetle species. It's a movie in a like a what do you call it, speed, in a fast speed, and the beetle coming in, nothing. This is maybe in a 30-second, but actually it might be a couple of hours. Anyway, sweet potato weevils. So we have two kinds of weevil. One is regular weevil, one is regular weevil. I and James, and including Emily, working in our project, variety screening project here for nematodes as well as these beetles. One is regular weevil, another is rough weevil. We don't know if we have rough weevil here on Kauai, but they are present. They are known to occur in Big Island. But even though we don't think we haven't seen rough weevil here, but we have seen the damage caused by rough weevil here in Kauai also, right? They make a tunnel in the tuber surface. They stay in the soil and make a tunnel, this kind of damage. And unlike in a regular weevil, what they do is they bore a hole, go inside, make a larvae, lay egg, and the egg will lay, and then they damage internally. Regular weevil damage internally, but the rough weevil damage external. That's why we cannot find the weevil. It's in the soil. So at some point, maybe we need to sample soil and try to find out if this rough weevil is here on Kauai as well. So leaf hoppers. So I put this slide because we have a hopperborne problem in papaya also. Not very common, but I can find this problem in a some form. So as I said also before, yeah, plant hopper damage in corn, making a hopperborne symptoms. And similarly in papaya also, they make a hopperborne symptoms in papaya. I think this problem might be more common in Big Island. I'm not sure on Oahu, but we don't have much of this problem here on Kauai. So a little bit of disease. So the fungal disease is mostly characterized by leaf spots. It's also called blight in some cases. And if it comes in the fruit, It's called fruit rot. And most of the case, so fungal disease, if it infects root, it causes root rot. Then we call root rot disease. If it infects stem, it splits stems and something oozing out from the stem, then we call it as a stem canker. And if it's infests fruit, rots fruit, we call fruit rot, and leaf spots are very common. So this is the late blight disease in tomato. You can, so same disease, when it infects leaf, we call leaf blight. So now it will, these are in the stem blight, and also the leaf blight symptom in the tomato fruit as well. It will look like this. So it infects all plant parts. Again, this is a very common disease here. Livi to east side onwards, very common disease in papaya. Phytophthora root rot. Again, we call Phytophthora root rot if there is a root rot. So sometimes you will see the stem, trunk of papaya very slippery. In the rainy season, it's very slippery. And if you closely look at the stem, you will see spores coming out from the trunk as well. So stem canker, then when it's a severe, it's the stem, the bark of the trunk will slip and something will ooze out, you will see. And when it infects papaya fruit, it's a papaya fruit rot, same disease. So we have another fungal disease and bacterial disease as well. So it's difficult to distinguish between fungal and bacterial disease sometimes. So this is my formula. If the leaf spots are circular, it's a fungal. If the leaf spots are angular, these are a little bit angular, yeah? It's not circular. So, and it has some kind of a margin in the leaf spot. It's a bacterial leaf spot. This is a hibiscus leaf spot. So also most of the, after there is a disease, most of the plant parts rot, fruit rot, stem rot, root rot, whatever rot. In bacterial disease, there is some stinky smell. And in fungal disease, not much smell. So if there is a very stinky smell, I usually call it might be bacterial, but not sure. But that's just to give an idea. Yeah. So this is a soft rot of banana. Lots of rain. There will be rotting of a trunk. And this is one of the very common disease in a ginger, bacterial wilt, caused by bacteria. So it would, the pseudo stem will rot, and when you cut the rhizome, you will see the brown circle in the center. And if you try to put in a clean glass of water, you will see, you don't need to move you just put and set aside for a minute or so, you will see the stream of bacteria coming down from the rhizome. That's how you confirm it's a bacterial wilt in our field level. We don't need to send whole samples to diagnostic lab. So same in a tomato also. Tomato also has a bacterial wilt. So just at the right time, around flowering stage, the tomato plants will wilt. The branches with the flower will wilt first. So then you suspect it's a bacterial wilt. Then you confirm, cut the stem, put it in a clean glass of water, and if you see the stream of bacteria coming down from the stem, it's a bacterial disease. So these are the, now I have some of the control methods, different control methods which we can implement as a IPM methods. So first of all, cultural control. The cultural control is the modification of normal practices to reduce the pest problem with the sanitation, crop rotation, trap cropping, using of resistant variety, altering irrigation and fertilization to make plants healthier so that they are resistant to some insect attack and also as a disease. I might have a little bit elaboration in the next slide, like a host plant resistance. So normally if there is a fruit fly problem, fruit fly likes smaller fruits than the larger fruit. So the larger big stick variety, other way around. Fruit fly likes smaller tomato fruits than the larger variety. So we might choose smaller tomato varieties like cherries to do fruit fly management. So this is another kind of managed control methods, which is physical or mechanical control methods, in which we exclude the insect by doing something, just like hand picking of Chinese rose beetle, using traps, permanent traps, which attracts only male, so that there is a netting disruption. We can use barriers, row covers, plastic house, bagging of fruit to control fruit fly, and also spraying of high pressure water to dislodge the insect. This is also useful to manage the Chinese rose beetle. And blower, vacuum to suck the insect. So this is a hand picking of insect in our home gardens. These are Chinese rose beetle, in our beans, and this is actually a rose plant. So what we can do is we can go at the dusk in the evening time with the torch. We can physically remove the Chinese rose beetle. So these are some examples of row covers to exclude the insect. Bagging of, this is a lychee, bagging of lychee, maybe for the roasting parakeet, and also for fruit flies, screen house to exclude insect. Another important method is the biological control. So we introduce biological control. There is three different kinds of biological control methods. Normally, what we do is we enhance the activity of natural enemies so that the pest population will go down. Three different kinds of biological control methods: classical, augmentative, and conservation. So in classical biological control is when a new pest is introduced in a new area. So for that insect, it would be an enemy-free zone. Only insect will come to new area, not their enemies. So it's an enemy-free zone. So in that kind of situation, that pest multiply rapidly and become invasive. So in that kind of scenario, what normally is the procedure is a few researchers will go to their native range and find the natural enemies of that pest and bring back to our place, and it will be in quarantine for several years. They do some host specificity test, and some non-target impacts. It will take maybe, I would say, 5, 6, 7 years, and then if it passed everything, there will be permit to release from maybe USDA, and it will be released so that the pest population will be under control. So as you can see, this is a photo of a researcher. So our old former county administrator, Rashad Manangar and Mark Wright, went to, this is one country in the South Africa. Can you say the country name, looking at this tree? Anyway, they went to their native range in South Africa for Erythrina gall wasps in Hawaii. So we have Erythrina gall wasps detected here and become very problematic. All the willy trees dying. So they went to this country in South Africa, found the natural enemies, came back to Hawaii, was in quarantine, did all kinds of tests. Then it was permitted to release, and it was released here when I was a graduate student, around 2008, 2009, 2010. It's established in all islands, and it's supposed to be that the gall wasp population is under control, and the willy tree may be reviving. I don't know after that. So that's the general procedure of classical biological control. So another is augmentative biological control, which we already have a pest it has a very specific natural enemy, and it's mastered in the laboratory and released as many times as you could, so that the population is lowered very much. So one good example is a trichogramma egg parasitoid. So I also did some research in this trichogramma to control corn earworm. Maybe I will have some slides in the next talk. So what you do is you mass rear and release in the cornfield, and the earworm population will go down. So in most of the cases, you can buy these natural enemies, just like you can buy ladybug, you can buy some other parasitoid, but we cannot. We cannot bring those parasites. There's lots of different companies in Canada and mainland USA, but it's difficult to bring those, bring those egg parasitoids or predators in Hawaii. So another method is the conservation. So the natural enemies are already there. So we conserve them we conserve and enhance their activity with supplementing floral resources, or with supplementing insectary plants, the plants that provides food and shelter for those natural enemies. So we mostly main crop with the flowering plants. So we can enhance the population of these sylphid fly, as you can see, and minor pirate bugs, and also parasitoids. So as a biological control agent, this is the scenario what it looks like. There is a pest population fluctuating. Yeah. So once you release a biological control agent, and it will tend down, and it will under the threshold level. And it's this is the example of classical biological control. So you release biological control in a small amount for only 1, 2, or until it's established. So it's only a few times. You don't need to release several times. A couple of times, 3, 4 times, until it's established. So once you release, the population will come down under the ETL. Then the plants needs to be happy if it is under ETL. So these are some of the photos of a parasitoid parasitizing an aphid body. Aphid, the larvae of the parasitoid will emerge inside the aphid body. This is inside the aphid body, larvae inside the aphid body, and the parasitized aphid will look like this. It's look like a metallic aphid. It's called mummified aphid. So some of the biological control agents, predators, ladybug, parasitoids, these are the parasitoid of aphids, and the pathogens, BT. Taxonomy, we have seen this photo, green lacewing, yeah, eggs. They lay eggs in the stalk and attached to the plant part. These are the larvae or predators, and they pupate, and 2 different kinds of manure. So sylphid fly, it's also called hover fly. Their larva is a predator. So egg, larvae, pupae, and adult. But larvae will feed the other small soft-bodied insect. Minute pirate bug, nymph. So the eggs hatch into nymph. Nymph will develop adult. Both nymph and adults are predators. So see, they have a stylet. So they extend stylet and punch into aphid body and suck all the juice. So you will get only chaffy aphid. So egg parasitoid, Trichogramma egg parasitoid, they lay egg inside the eggs of other pest insect. Same thing happens, the eggs will hatch into egg. The eggs will turn black, then we call it parasitized. Then the parasitoid will come out, not the insect. Braconid parasitoids are larval parasitoid. They lay egg.
Sorry.
Hi. Okay, I will finish this presentation. I have only maybe a couple of slides left. This is a braconid parasitoid. This is larval parasitoid. They put the eggs inside the larval body, hatch the larva. Then after several instar, maybe 3 instar, come out from the larval body and pupate. Pupate like this. These are the pupae of the parasitoid. There is one movie, one parasitoid attacking a aphid, and put the egg inside the body and parasitize, but we cannot see now. So some of the successful biological control projects here in Hawaii: sweet potato hornworm, citrus swallowtail, banana skipper, sugarcane repeat rotor. So these are some of the successful biological control. They bring the biological control agent released here, and the main good thing is you might not see these insects here anymore. So the question is, is biological control agent, biological control method is dangerous? So a long time ago, there was a problem in a sugarcane plantation with the rats. And at that period of time, growers, most of the growers, what they thought is if we bring the mongoose, they will eat all the rats, and there will be no rats in Hawaii, mostly in the sugarcane plantation. So what happened is this mongoose is durable. They roam around and hunt in the daytime. The rat is a nocturnal. So what happened is they cannot control rat, and these mongoose became invasive and also hunt our native bird species. So that's the reason it's very difficult to implement or get permit to import biological controls. But I think now we don't have that kind of scenario. That without understanding the biology and behavior of the natural enemy, we don't bring biological control now. But nowadays, with one of these examples, it's always difficult to import any biological control agent and release here. So it takes numbers of years to bring the biological control agent and release. So chemical control, so these are some of the advantages and disadvantages. Effective, cheap, yeah, simple to use, but it has some kind of these negative impacts. So always read the label and follow the label. The different classes of chemicals, I a little bit touched upon these organochlorides, organophosphates, carbonates in the early era. So the pyreters, neonicotinoids, and IGR are the little bit newer kind of chemicals. I think organochlorines and organophosphates is already banned here in the United States. So I also said all the insect pests here in Hawaii are invasive. So where did they came from? See, cosmopolitan, from everywhere else. 36 of them came from somewhere. Okay, it's cosmopolitan. It's found in all parts of island. From Asia, 21 pest species came to Hawaii. From North America, 11, so on. So whatever insect pests we have here in Hawaii, all invasive. So other invasive species extension is what we do is we talk about detection, eradication, something. Several years, lots of funding, lots of research. Then once that stage is passed, then it becomes pest. So whatever pest we have at one point of time, they are invasive. If they are not invasive, if we were able to eradicate, we don't talk right now. Yeah, it's gone. So whatever we have here, it's all invasive at the time. Invasive. So the point of saying that is controlling invasive species is the first line of defense. So if you have some new detection, we need to act. We need to do research. We need to do lots of different kinds of things to limit their spread. Try to eradicate. Otherwise, we will do the same thing. We will count the numbers of insects coming out from several different countries, and this become, then we thought it become pest. And also a little bit of politics. If we go outside of Hawaii to mainland, I think they will check our baggage 2 times. One in here, yeah, and one when we go to gate. They will check our baggage thoroughly at the gate also. So when we come back from mainland, what we do, we just fill out this form, small form. So that's a little kind of bias. So what does it tell? If something come out, new thing come out in Hawaii, it's okay, but we cannot take to other United States, mainland United States. This is a little bit biased. Just wanted to point out, and just like as I said, be alert. These are the now current invasive species. Coconut rhinoceros is only on Oahu, but not other island. Brown marmorated stink bug, not in Hawaii. Very much problematic in some northwest United States. I think it's spread across some number of states now. Little fire ant, we said we don't have, but now and then we always detect with mostly DOA and KISS guys. So be alert. So if we can stop the insect invasion at this stage, it's things to do. But once we cross this, it becomes pest, and we need to talk. We need to talk, and we need to control that insect for a long period of time. And thank you. Okay, I finished this too.
So we have some questions before we go to a break. So from the chat, there was someone asking, are there good, inexpensive methods for doing things like the traps or some of the other scouting things? Any sort of hacks people use for those?
So normally traps will be, so light traps are light traps, just we need a light. So normally nocturnal, most of the insects are nocturnal, they attract to light. So light traps are inexpensive. As I can see one of the in a street art site with the one master gardener developed light trap for Chinese rose beetle. So Chinese rose beetle doesn't like a bright light, but they like a dim light. So I think one of the master gardeners a long time ago, maybe 5-7 years ago, he or she developed a light trap with the dim light. So light traps are normally inexpensive. And another thing, the traps will be associated with mostly lures. If it is a pheromone lure, we can make our own traps out of box or bottles or in a different kind, but lure needs to be there. The fruit fly has a specific lure, specific species, specific lure. Melon fly like Q lure. Other Bacterial dorsalis, the one in the fruit trees, they like metalluginal. So another thing is most of the insect like fermented smell. Fermented smell, so we can replace lures with the some kind of fermented product, fruit, or we can make some kind of a sugar added with yeast. So whatever insect which likes fermented smell we can trap. So I think those can be inexpensive traps. One with the light. So take a container or a bottle or whatever. One with the light and one with the fermented product. So fruit fly are also attracted to fermented product. Lots of ambrosia beetles are also attracted to fermented product. Mosquito is also attracted to fermented product. Yeah, lots. But it will be less efficient than the specific lure.
Another question about how do you decide what the thresholds are for a treatment level versus the keep sampling level? I know you got into this a little bit, but how did they kind of set those standards?
So I give a couple of examples with the banana bunchy top virus and the coffee berry borer. So I think they did economic analysis based on the damage or the symptomatic virus plant. So I think it's a long process. It takes lots of research. It might take 3-4 years. So I showed that 2 scenarios. with the coffee berry borer and the banana bunchy top. That's it's a couple of graduate students involving in the banana bunchy top virus. And I don't know, I think that the whole faculty team were there in a, I think it's some kind of an importation from the South America as well, the hereditary sampling, and they did their economic analysis for Hawaii itself. So it's a difficult question. So ETL is, as I said, decision support system. Yeah, for us, if we need to decide by ourselves. If we think this damage level will make my income less, then we decide to spray. That's how I think we need to do it. Not as a first sight of insect spray.
The third part, is that a statewide presentation?
We can, yeah. Oh, it should be, yeah.
Okay.
Whatever.
So we'll do another maybe 5-minute break this time, and then come back, and we have one or
Okay,
two
thank
more
you.
questions we'll do with that. So 11:06.
Ooh. Drink water. So third presentation what you did?
Click on the three dots and then click hide floating meeting controls.
Okay, welcome back.
Maybe we address a few quick questions, if that's all right.
Yes.
So someone had asked about an update on the biological control for strawberry guava. I know it's more weed than.
There is a scale insect. Is that? I don't remember now. So I think it was a long time ago, the biological control agent of strawberry guava was implemented. It was from a big island. I don't remember this one. yeah.
We'll have to talk on weeds later, so we can ask the expert for that.
So the scientist, I cannot remember names. He's from volcano. Everything was done in the volcano, and the biological control agent was released. And as I said, it's a classical biological control method. So what they will do is they will release for only fewer times, not all the time. They will come to Kauai, release, come to Big Island, they are from Big Island, release, Oahu, release. Once they think it's established, then that stop. So the strawberry guava will be under check after that. That's the principle of classical biological control. I think it's already done. It was long time before, maybe 2010, 12 or something.
Another question about does silver reflective mulch help keep insects away?
Yeah, mostly aphids, whitefly, and thrips.
I've never heard of what is silver?
Silver reflective mulch?
Tinsel?
Yeah.
Silver reflective mulch. It's so normally what do I know about this is reflective mulches are used to control virus diseases transmitted by either aphids, thrips, or whiteflies. And I don't know in Hawaii they have used silver reflective mulch here. In California, yes. So the reflective mulch is the paper mulch. We have a plastic mulch, black plastic mulch. Similar to that, it will be silver. It's shiny. So it's a shiny reflective mulch. So the sunlight, white light will come, then reflect some other wavelength of light. That will deter some specific group of insects.
That they're lighting at the underside of the leaves.
The wavelength after reflection will be different, so that some specific group of insects will not like it and doesn't come too close to that area.
And I think it's not kind of a shredded mulch like we think of. It's normally like a panel, like a plastic sheet.
So it's a plastic sheet. There is another kind of mulch also. You spray in the soil also. That will give. There is a.
Oh.
Yeah, not in Hawaii, but I have seen in different states. Yeah.
Excellent. Anyway.
So okay, so welcome back. This is the third slides. This is a kind of the one before. These are mostly kind of a technical thing. This is a little bit more practical that I have gained some knowledge during my 3 years work as an extension agent for invasive species. This is me spraying BT on kale to control diamondback moth. It doesn't die. That's why I spray it every other day. So same thing. So just wanted to talk a little bit about crucifer pests, lots of caterpillar pests, and also one stink bug. Among these, hopefully everybody is familiar with these insects. Cabbage butterfly, diamondback moth, cabbage webworm, and bagada bug. I will go through one at a time. You can see what I have found is diamondback moth is not very common here. The more common is cabbage looper. So this is the larva of the cabbage looper. When they walk, they loop their larval body. So it looks like that. That's how they are called cabbage root rot. They have a silken cocoon and nocturnal adult. And you can see the eggs in the leaves, circular, round, scatter. I think I have something. Okay. So cabbage webworm is not another insect, not common here in Kauai. I think they have problem with cabbage webworm on Maui, not very common in other islands, only problematic on Maui. I have seen this webworm, cabbage webworm on this island, just like a very few here and there. So the larvae looks like this. Sometimes it's embedded in the leaf tissue. They make some kind of a space to hide themselves, and not very damaging here also. But this is from a photo from a Kylie Tavares. So it can make cabbage like this. Again, nocturnal. I haven't seen eggs. Another very common pest, cabbage butterfly. You can see these barrel-shaped eggs with your eyes. The larva hatch. The larva of the looks very beautiful. They have a fuzzy, what do you call it? Lots of hair, velvet. It's a very good in touching. So this can grow very big and fat, and they pupate into this shape. It's called chrysalis. And adults are diurnal. You can see these butterflies flying around your crucifer field. So this is supposed to be very damaging pest, but I haven't seen this pest that problematic as other states do. Eyes are not seen by the eyes, needs microscope. These are the larvae. So how do you know? So one of the distinguishing feature of this diamondback moth larvae is a tail. Tail is biforked, and also if you touch this larvae, they will wiggle. The other larvae doesn't wiggle, but this will wiggle. And they feed on these leaf tissue, but leaving this epidermal layer. The pupa is the silken cocoon, and the adults looks like that. When they fold their wings, it makes 3 to 4 diamond shape on their back. That's why they are called diamondback moth. So as I said, this is normally called caterpillar complex in crucifers, not only diamondback moth, but the diamondback moth is the one which is very difficult to control because of the resistance developed to different classes of insecticide. So as an IPM, we can implement different control methods: physical, biological control, cultural, and chemical. So for specifically diamondback moth, they hang out, they are also diurnal. They hang out around the evening time. And you can see the diamondback moth flying around here and there, one plant to another in the evening time. So the sprinkler irrigation will help them to colonize, to stay in the one plant and oviposit. So if you have a sprinkler irrigation head in your crucifer field, that helps minimizing the oviposition of diamondback moth. So another thing is a vacuuming diamondback moth adult. So this is a leaf blower. You can make this leaf blower into some kind of vacuum suction also by filtering into different way. So what you do is in the afternoon time, you suck the adult diamondback moth. So that's another way to do physical, as I say, physical control is physically removing the insect, yeah. So another thing is matting disruption. We have a pheromone for diamondback moth. We put the traps with the pheromone that will attract and trap only males. So there will be only female diamondback moth. They will lay eggs, but the eggs will be unfertilized. So these are some options we have. So biological control, we have predators, lots of different predators, parasitoids, and the pathogen. Predators and parasitoids in this case is not that effective. They are not specific predators that only eats diamondback moth. And in case of parasitoids, we have one species, Clotaceae plutili. So what they do is they only parasitize late instar larvae. Late instar means only third instar. After third instar, the third instar of the diamondback moth is when they almost did all the damage. So they already did the damage, then the parasitoid come, then parasitize. So these are this is the larvae of a parasitized diamondback moth, larval parasitoids. The larvae develop and they pupate outside. So my meaning is to say is even the parasitoids are effective, but they attack after whatever they damage has already done. So pathogen is BD. They have different strains of BD. It's found in soil, but it has been commercially formulated and used to control mostly Lepidoptera larvae. So these are effective when it's correctly used. So in cultural control, the idea is to manipulate the growing system. So breaking the crop cycle, if you grow crucifers all the time, season after season, the brassica will stay. And the one thing we can do is a crop rotation, break the cycle of the insect. Another thing is, in case of diamondback moth, they like crucifers. They are very specific to crucifer because of crucifers plants have some levels of glucosinolates. So they respond to levels of glucosinolates. That's why they only attack crucifers. Also, in crucifers, different varieties of crucifers, different kinds of crucifers. Also, different varieties of crucifers have different levels of that fuel, mediated by levels of glucosinol. So they have a differential preference of crops. They like collard more than cauliflower. They like kale more than something else. That's why the trap cropping may play the role in controlling the diamondback. The idea is to draw the insect population in a trap crop and kill them. So this is my little bit of experience where I was in Missouri. I did a trap cropping research for diamondback moth. What I found is kale is more attractive to diamondback moth, and collard is attracted to cabbage butterfly. We have lots of different caterpillars here, so that's what I found. The diamondback moth is attractive. I have lots of different cruciferous crops. So this is choice taste and no choice taste kind of research. So diamondback moth is attracted to kale, and cabbage butterfly to collard. So what I did is I want both insect to control. Yeah, I mixed the trap crop. So mixing up trap crops and spraying insecticide in the trap crops to control. So normally this is a thumb up rule. 10% trapping is economic. If you put more trap crops, it might be more effective, but you lose money by devoting your land to trap crops. These are the two different systems of trap crops. One is you put the trap crops in the border, and another is perimeter trap cropping. So I just mentioned mixture of trap crops. So the first thing I did was mixture of trap crops, research and mixture of trap crops. And another, then as we developed the idea, then we put mixture of trap crops with the insect-free plants. So idea is trap crops, two mixture of trap crops will attract two different kinds of insect, and insect-free plants will enhance the activity of natural enemies. So I have these sweet alyssum deal fennel basil as the attractive insect-free plant. So did some kind of research, but not great results. That's why I did not have result here. So now also here, I collaborated with Kunhui Wang in trap cropping research. And this time we have a trap crop in cabbage, but we are using entomopathogenic nematodes to control insects, which is attracted in the trap crops. So basically, it's for the organic growers. You use trap crops and use internal pathogenic nematodes, which kills Lepidoptera larvae in the trap crop.
So it's nematodes.
Nematodes, yeah, it's an internal that kill insect. Internal pathogenic nematodes will cause diseases in the insect larvae and rapidly kill. So again, chemical control, as I said, I was spraying BT. So the farmer here in Lihui told me, I spray every other day BT, I spray interest every other day, but I cannot control diamondback moth. So the CTAR extension has a statewide program of insect resistant management for diamondback moth headed by Robin Simabuku. Now I think Rosemary is taking the lead of that. I think that talk will be here in Master Garden.
I'm not sure if
So if not, we have some results from that insect resistant management here on Kauai. As I said, we don't have a diamondback moth as a major problem, but still we did. So what we did is we collected the population of diamondback moth from Lihui. Only it's a little bit problematic in Lihui, and we took it to Maui to do a bioassay with different classes of insecticide. So diamondback moth here in Kauai is not killed by interest, BT, and radiant. So we took the population, reared them out, and we applied these chemicals. In the x-axis, there's a different chemicals, different classes of chemicals. You can see the names, very big new names, and y-axis, mortality percentage. As you can see, radiant control only 13.8%. Radiant yielded only 13.8% mortality. Movento didn't kill. So out of these insecticide, so they are resistant to radiant and also Movento. And among these, Exiril, Thorac, and Proclaim is very effective. And other insecticide like Rimon, Coragen, and Alvan is very useful to control diamondback moth. Then what we did after this result, me and Emily, so this is all Thai growers. So me and Emily communicated the Thai growers that's spread around. We are on 14 farms. And so what we recommended is this. So use new insecticide other than radiant interest and BT. So in Kauai, they can buy only Thorac. Out of those 6 or 7 insecticides, only Thorac is available here in Kauai. So what they can do is they can spray Thorac. I didn't say Thorac, but they can spray new insecticide out of those lists. So they can spray 2 spray at weekly interval. You spray this week, you spray next week. Spraying weekly conjugative time is to kill all the resistant population at a time. So first spray, maybe they only 90% of insect is killed. Second spray, we need to kill 100%. So in a month, so 2 spray, cannot spray that insecticide anymore in that cropping season and that year. So if you need to spray anything else, if you see, if you think, if you feel there is still diamondback moth, you need to spray, it's called filler spray. You can use one of these, BT. There are 2 strains of BT here available, Zentari or Diplo. So if you need to spray other insecticide in a subsequent week of that month, you can choose one of these, or maybe you can repeat also, but you can spray this in a 3rd or 4th week. Then, if the diamondback moth is still a problem in next month, you need to choose new insecticide, not Thorac, and not the old insect. So again, same cycle, 2 sprays of new insecticide fill in by PT in that month. So what we assume is after 2 months of spray, we assume, or it should be, the resistant population of diamond black moth is eradicated there. So then after 2 months, if you see again diamond black moth, you can use a less effective insecticide. You can go back to radium, interest, and BT alone. So that was our communication with high growers. And what happened is after they used Chlorotic, there is no diamond black moth after that one. So for the second year for the buyer, they say we cannot find diamond black moth. So this strategy is very useful, even we don't have lots of insecticide available. For a statewide resistant management project, Robin Siambaku now, lots of collaborators in different islands, what they do is they recommend 6 different insecticides for 6 months. And all the growers need to use same insecticide on that month. That's 2 spray at weekly interval in the beginning. Then if they need to spray, they can spray only BT. Otherwise, no spray. Then next month, next insecticide. So they recommend same 6 different insecticides for 6 months. And specifically it's for Maui, and I think it's Oahu and Big Island also. And it needs to be strictly followed. Otherwise, you spray X, the other spray Y, it doesn't work if the 2 farms are close together. So that's the insect resistant management statewide project, and it's very successful up until now. Oh, I have a cabbage webworm. So this is occasional pest. So I was talking about occasional pests, secondary pests also, yeah. So if we don't care of this occasional pest, also this can be a primary pest as well in the future. Bagrada bug, this is listed as an invasive. It's invasive in Maui. Lots of problem in Maui. Not here. Not here. Only occasional pest here on Kauai. I have seen this pest in the daikon and other kai choy, pak choy kind of crops. Only once in a while here and there. Lots of different host plant. So just to show you, keep an eye on this pest as well. Bagrada bug, not very much here, but it can be primary pest if it will establish here. It hasn't established well here. So feeding damage of Bagrada bug. These are some of the feeding damage. I haven't seen these damage. This is from STOA publication. Okay, let's move to some new pest. It's not new pest. Yes.
Just as a comment on Bagrada bug, I know we haven't found it here, but for folks on the neighbor islands, we've started to notice it shifting to other crops too. So I saw it on sweet potato when I was on Big Island.
Let me see.
So just keep an eye out for it, you know, even if you're not growing some of the more known.
So I have alternative host as a corn, cucumber, okra, sugarcane, papaya, potato, maybe sweet potato. Potato is sweet potato as well. So yeah, this is a problem in Maui especially. It can be here too. I will not talk too much about cucurbit place. Fruit fly, everybody knows. There is a very good pickleworm also. Lots of people confuse between fruit fly and pickleworm. Mite fly aphid. So aphid vectors, another virus disease called papaya wing spot virus. Lots of work has been done in papaya wing spot virus, and they have GMO papaya, which is resistant to papaya wing spot virus. Fortunately, we don't have this virus here on this island, so I will not be talking about this. Only a little bit about fruit fly, which is also melon fly. So as we all know, from a long time ago, there is a IPM recommendation, which is called as EG 123 melon fly control, which is field sanitation, monitoring traps, and to sanitize. You need to pick up the infested food from the field. Yeah? Don't leave the food. And monitoring traps means you put the traps with a certain specific lure, and then protein spray. You can read this publication, lots of publication in the CTAR website. I will not go detail, but there's a little bit of new approaches in the fruit fly control as well. So this is work done by our colleague Rosemary Gutierrez in Maui to control melon fruit fly. It came out as an impact statement in a CTAR. So what the approach is to find out the roosting side of fruit fly. You don't apply traps or insecticide in the crop itself. You try to find out, mostly fruit fly will hang out in somewhere else in the daytime, in the shady areas. In the mango tree, in the avocado tree, in the castor bean patches, or in somewhere in the city area. So the first step is to find out the roosting site using these torula yeast protein traps in a multi-lure. Torula yeast is, I think, it's a protein source. It came out in tablets. And what they do is they put 3 tablets in a 20 gram of water and put it in this multi-lure trap. So first thing is to try to find out the roosting site. Once you have a roosting site, you can have another trap. This is another trap. You have some kind of a paper board impregnated with it's called amulet seal. Amulet is maybe treadmill. Seal is the cure again to attract a melon fly. So what they do is in this cardboard or some kind of a spongy paper, they inoculate cure with in some insecticide. The insecticide is Phytophthora, and it's proven it's very effective. It can attract male from 130 feet, and it works up until 5 months in the field. So a little bit new approaches than what we used to hear in a fruit fly IPM. So if you are interested, you can read this paper in this website. Pickleworm. So normally, if you have a fruit fly infestation in zucchini cucumbers, you will have a mushy leaves, the fruit tissues, yeah. But in pickleworm, you will see a clear or circular form. Clear circular form. So that's how you know it's a pickleworm damage. If you put this whatever cucumber, zucchini in the water, you immerse in the water, then the pickleworm will come out. That's how you test if it is a pickleworm. So normally the female of this nocturnal moth will lay egg in the flower, and the egg hatch, and they will approach developing fruit underneath the flower piece if it is a female flower. So whatever management practice needs to do, we have a small window, flowering period. I don't know if it is 10 days in the cookies or a little bit more than 10 days. So whatever we need to spray is in the flowering stage, 10, 12, 15 days. It depends upon the crop. So zucchini might have only 10 days. So whatever we need to do in a flowering stage. Your spray needs to be directed in the flower and make them wet nicely. You can spray BT, interest, and gradient. So same problem as with the diamondback moth also. If you spray too much, the BT will not kill. So up until now, this is not serious resistant pest as diamondback moth, but if you can spray at right stage in the right manner, you can control pickleball. If you cannot control with BT interest, and you will go to another, maybe thorax. So a little bit about corn pest, 3 different pests: earworm, plant hopper, and thrips. Nocturnal, adult will look like this, and they will lay egg in the cells. So you will see the eggs on the cells. And the larvae, the color of larvae is different. If they eat green tissue, they look like green, and if they eat some kind of a, silks or some other grains, they will look like a reddish. That's how, so they egg on the silk as they tunnel through the silk channel, and approach to grain. So, so monitoring is important. So how you do monitoring? So it has a, I showed this trap before also, yeah, inverted funnel trap, and it has a lure for Helicoverpa. Corn earworm is a Helicoverpa armiger, and lure is called helilure. So the idea is to know the first incidence of Helicoverpa, the corn earworm. So it's not to collect trap the insect, but to get the indication of presence of earworm adult, or first incidence of earworm adult. As soon as you get the adults here, what you will do, you will go to your cornfield and look at the corn cells.
Brendan, you might have to turn that way and project, just because in the past it's been hard to hear in the back of the room.
Okay. So once it's something is trapped in the helilure trap, so the idea is to go monitor corn cells. So you see some eggs in the corn cell. So in the worst scenario, what I have seen is you pick the corn ear, and if you set the corn ear, you will find that some eggs dropping down from the corn cell. So in this scenario also, you have to focus whatever management practice is in the corn selfing stage. Might be only 10 days. You have only 10 days, and whatever you spray will be need to direct it towards the corn cell. But we have lots of other management options also. So as I'm talking about corn, I remember James Brubaker. I think he passed away only a few days ago. I saw an email yesterday. So James Brubaker is an eminent scientist in corn breeding. Whatever corn lines developed by CETAR is all by, I think, James Brubaker or his associated student. So yeah, great, hats off to us. So the thing I'm talking about, whatever James Brubaker or CETAR developed corn, it has a narrow silk channel. The silk channel is a longer, and it's a narrow silk channel. And the idea is the corn earworm larvae. So I think James Brubaker used to tell about how many what do you call it? Husk cover. There is 14 husk cover or 16 husk covers. I think the breeders can manipulate those husk cover also, yeah, right? The 16 or 14 they can manipulate. They need 16 husk cover so that they can do something. So they manipulate their husk cover also. So whatever CETAR released corn lines, it has a narrow silk channel and a tight husk cover, so that it's proven like we don't need anything to control earworm. So what happening is corn earworm is carnivorous. So there is a eggs on the silk channel they have, and at the beginning they feed on each other. So only few larvae or one or two larvae will be successful in getting to silk channel and feed on the kernels later on. So only in this corn varieties, chopping off the tip is recommended. So you chop off the tip, then whatever you have is free of corn earworm. So no insecticide treatment is recommended for corn, CETAR developed corn variety. So there is a corn earworm, no problem. They feed on each other. No, they just fight with each other, and one or few will go inside the silk channel. Maybe only a few percentage of them will eat the kernels. So only you can, there is a larvae in the chopped up this, and it's marked. The chopped up corn here is marked. So if the other varieties of corn from mainland or other parts of world is they normally try to develop a blunt, like a parallel corn here, so that they have lots of kernel in a cup. It's called cup, yeah. So they try to get lots of kernel out of one single corn. So that's what happened is the tip of the ear is already exposed to earworm, and they will go way deep inside to the ear and do real damage. So this is also a part of my study, during my study. So I try to compare conservation biological control method with the augmentative. So this is the mainland corn variety with the parallel curve here with the exposed tip. Super sweet, it's called Passion. The variety name is Passion. So the main idea is to intercrop with the sun hemp. The main purpose of selecting sun hemp is sun hemp flower gets these eggs from the lysine butterfly, and which is usually gets parasitized by Trichogamma species. So by providing these resources, sun hemp row in between the corn rows is to provide the sources of natural release. As soon as they grow, they already have a parasitized egg, and as soon as they attain certain stage, there is already Trichogamma in that area. So they parasitize the earworm. So another method is augmentative biological control method. I have a parasitoid Trichogamma pretiosium. So what I did is I multiplied them in the laboratory and released in the field, in the corn field. That's my whole other treatment. So normally the parasitized eggs will be glued in the piece of, this is a sticky note, yeah, and it's underneath the cup, some kind of cup, and it's hanged in the corn plants. So the release rates 100,000 and 150,000. So Trichogamma per hectare, twice in different stages, 40% silking stage at 5%. Then I compare the rate of parasitism in those two treatments. So green line, so normally in x-axis is that's the days after planting, it's the silking time. Y-axis is a percentage parasitism of corn earworm egg. So you can see this is a corn monoculture green, yeah? And yellow line is the rate of parasitization in a conservation biological control method, and red is the one with the Trichogamma release plots. Very effective, releasing Trichogamma augmentatively, increasing the population of Trichogamma rapidly in some period of time, at the right time, is very effective in controlling corn earworm here in Hawaii. Another pest of corn earworm, not corn, is a corn plant hopper. So we little bit talk about this. Yeah? These are the symptoms. So again, our all CTAR release corn varieties are Hawaiian super sweet, pure corn inbred are resistant in MMP. I think there is a gene called NBG, which is resistant to maize mosaic virus. And I think there is some technique that Brubaker and company, they incorporated that gene into our Hawaiian varieties and release lots of resistant varieties for MMP. So another pest and disease, maize chlorotic mottle virus transmitted by corn thrips. This was first detected here on Kauai in 2004. And it is transmitted by these thrips. It's not regular thrips which we found in a flower and in the silks. In the silks of corn, we found different thrips, but these are found in the young growth. What do you call it? Leaf group. What is this place? So leaf sheet. You will find only these strips under the leaf seed, and this corn chlorotic mottle virus can be eradicated, or it was eradicated here in Kauai by pioneer. It was sown up in pioneer that time by strictly following in the summertime. So if you don't grow corn for some period of time, 3-4 months, so you can eradicate this disease, and it's recommended. Otherwise, you get the problem now and then again. So this disease is here. This will show up and go, Oahu, every place. But we can control this disease also. Needs strict following. So, little bit about papaya, because papaya is popularly grown here on Kauai and maybe on all the other islands, known as a farmer, larger scale, small scale, and also in backyard. Yeah, so I have a little bit on insect and diseases. So these are the insects scale. White peach scale is common in all island, and there is a couple of other insect which is kind of new, detected a few years ago. I don't remember the name, but we detected this new scale here on Kauai in 2019. It's called Oriental Yellow Scale. I can say it's totally in control in one of the farm at Kekaha. We kind of eradicated, but it showed up a little bit later also, but still in control. You can see only few. If you go tree by tree, you can see only few. So in total, we have 4 different kinds of scale insect in papaya, but white peach scale is common, but we have oriental yellow scale also. But just to show you, you can keep your eyes open for this scale also, because these are the like a sporadic. This can be transferred by a year current. So normally, what is this? Is scale is the outer covering. The actual scale insect is under this cover. So what happens is the scale insect will lay eggs. The juveniles will crawl out, and they will be dispersed by wind current. So they can be easily spread around in the area. But we are able to restrict the spread of this scale. Another insect is a papaya mealybug. Very much big problem maybe in a big island, not much here. So these insects are normally suppressed by their natural enemies. I think the scale insect has one coccinated predator and one parasitoid. Similarly, they have a couple of different parasitoids in the mealybug. Normally, they are in a check with the naturally occurring parasitoid, but if it is out of control or we think if it is above economic threshold, we can use petroleum oil. It's very useful to control these insects with the oil. If not, we have another little bit useful insecticide. We also a little bit talk about IGR, insect growth regulator. I said this is insect growth inhibitor. This chemical buprophagine will inhibit their growth and kill it without making them molt. Normally, it doesn't allow insect to molt, so their growth is restricted in a small that area. So if not, we have a systemic insecticide, which should be effective than all of these chemicals. So a little bit of disease, black spot of papaya. So normally it appears in the leaves like this. We normally ignore, and it appears like in the fruit. Which also it can be sold in the farmers market or something like that. When we saw this, then it will be alarming. So normally spraying of, so this is very common disease. You need to spray fungicide at this stage, not this and this. So a couple of photos of papaya. Maybe you can recognize this. What will be this? This is some later development, but these spots have a concentric circle. Yeah, this is called anthracnose disease of papaya. Huh? Fungus, yeah. Fungus disease. This is the anthracnose disease. It's mainly appear once you harvest papaya and ready for sale. Next day it will appear. In the field, you will only see small scratch or blemishes. So normally, I put this papaya as an example because all these diseases will appear in fruit crops as well as in vegetables. So what is this then? The other form. So this is the mite damage in papaya, not disease. Another disease, very common disease in all different kinds of fruit, as well as vegetables, or free mildew. So these all kinds of disease appear maybe in a time. So there is in a favorable environment like this, there is a disease complex. So what do we do? So we cannot alter our this situation, weather condition here. So we have different disease appearing at a time. So we have another disease. This is a root rot of papaya, which is another main disease. So we spray fungicide. Normally we need to spray fungicide. So what is recommended for papaya and other crops, we need to spray fungicide every month. If situation is like this, it's recommended to spray fungicide at 2 weeks interval. So normally one fungicide is effective for one disease and other fungicide is effective for disease. So how do we plan to spray? How do we make our plans to spray fungicide? Suppose I saw a black spot. We normally say cocci, no cocci. Mite is effective for black spot. If we have a anthracnose, cocci is effective, maybe cocci. This is not my recommendation, but just to give you an example. I'm not recommending if you see this, you do this, but just an example. Powdery mildew, abound and rally. It's in the label, so abound and rally. If it is a root rot still come. So root rot, I have discussed before slide also here. So root rot, maybe cocci, bravo, lots of different kinds of fungicide. So what to spray, how to spray, if we need to mix the fungicide. So we need to rotate the fungicide, and we can mix the fungicide as well. So normally the label will say, if you can mix with this, that, but they will not say, yeah, you can mix cocci with Mite. So it's very challenging. So we need an experience, we need to try by ourselves. So normally what goes through, or what I have done in the past, if I can mix this cocci with Mite. So what I do is I do some kind of a little bit of small test. Dilute, mix with cocci with Mite. If the color doesn't change, if there is no fumes coming around, if there is no vigorous reaction, I'm not recommending to do it. But that's how we need to experiment, learn, and try.
We have about 5 minutes left before doing.
Okay. Root rot of papaya, I already talked about this. So we need to plant papaya in it's already 1 hour? Okay. So yeah, so I did a small experiment for root rot, trying one new fungicide, which is systemic, metalaxyl. It's called metalaxyl. Trade name is Binomial Gold. I have a foliar spray, soil drains, and mixed with cocci. That's why I'm talking about it. Mixing up two fungicide. So I haven't taken the data, but the plants looks good and normal. These are the bacterial disease. I also talked a little bit earlier, a little bit more about other diseases, a little bit lax scale. Not here on Kauai. I think when I was working with Dr. Cheng, I used to come here to survey this insect, have found once in one departmental store. And this was on Oahu, on Maui. So the main thing is it has 80 host plant species belonging to 34 families, and 15 of them are native. And if Dr. Singh will be talking in a later edition of MJ Master Gardener training, he will talk more about his using of systemic insecticide as soil drains and trunk injection. Avocado lace bug, little bit hot news in Big Island. Lots of now this insect is spreading around Big Island. So I think there are lots of CTAR faculties have interest in doing research in this insect. And for now, I think there is only we can recommend is an insecticidal soap or some kind of a, mineral oil to control. I think it's effective for controlling this insect. Two-line spittle bug problem in Big Island. I think it's not new in here, but not problem here. they like to eat like a pasture grasses, kikiri and pangola, and they remove this habitat, and it's overtaken by the invasive species, invasive grasses, and there's no control methods yet. The only control method is extensive grazing of livestock in that area, so that they can eat infestation and also insect. Little bit response. I would like to talk about this, but I will not. But for CBD, there is a little bit more research on biological control. There is a predator called square-necked rain beetle. It can be easily reared in the broken grains and corn flour. So what they recommend, that's a research done by Peter Follett and Andrea Kawabata. So what they have a container with the corn broken grains, and they inoculate a little bit of this beetle and leave it in the coffee field. They multiply, go out and find the coffee beetle or larvae, pupae, and try to remove from the area. More research on coffee beetle, biological control agent. This is a Phymasticus coffeei. It's an adult parasitoid, still in quarantine in Big Island USDA facility. I hope it will be released soon, then we can release in our coffee field. It's under the non-target impacts study. And up until now, it doesn't attack our native ambrosia beetles. So hopefully it will be, there will be permit for releasing them soon. And there is one of the main problem I have heard as an extension agent is lots of ambrosia beetle in a different. So this is ambrosia beetle in a ohia box. They mainly attack in the main trunk. So ambrosia beetle in the avocado, one ambrosia beetle which is called Xylosandrus crassiculatus. They like to attack in the young seedling saplings and the younger plants. See, so you can, if you closely look at this, you can see a toothpick. So what they do is they bore a hole, they tunnel, eat, and this is the frass coming out from the tunnel. Very difficult to control. Once they attack the main trunk, the tree will eventually die. So we need to replace the tree. The other ambrosia beetle, which is a regular ambrosia beetle, we call mostly it's a coffee bark. No, black twig borer. So these are the damage caused by the black twig borer. They basically find the twigs, branches, not very thick diameter. They try to find only twigs, so they damage in the everywhere. They have lots of those species. This is a mango tree, this is a longan tree, and there is no way to control. Cannot spray or cannot inject insecticide in the fruit-bearing trees here on Hawaii. So the only thing we can do is either cut the branches and safely dispose, or cut the trees and plant new one. Okay, yeah.
Maybe at this point, we'll switch over to answering
I'm
a few
done.
questions.
I'm done. So there is a new discovery. So there is a new discovery in the coffee bark beetles. The one guy who works with I forgot his name. Very difficult to remember English name for me. There is one guy who is a graduate student with the Mark White. He has discovered lots of parasitoids of bark beetles. You can see YouTube video here. Otherwise, I'm going to play it right now. And this is the last slide. Thank you.
Perfect.
Yes. Thank you very much.
I think
So this is a very interesting YouTube. No, don't miss it.
And I'll put all the videos here on the CMS site and also upload them to the YouTube playlist. We also had some questions in the chat about some of the links that Rashad shared. And so I'll be putting those under his name on the CMS site where the slides are available as well. I think we're pretty much out of time. I don't see questions in the chat that haven't been addressed. So I think at this point, we'll switch over to the individual programs to do their things. Hawaii folks, I know there's folks here, but for the folks that are on